Um, I think there's people are still joining, but well, it's my pleasure to introduce today's seminar speaker, Dr. Alejandro Tena. Alejandro, he's a specialist in biological control and integrated pest management at the Plant Protection and Biotechnology Center of the Valencian Institute of Agriculture Research, IVIA. And he received his bachelor's in agricultural engineer from the Polytechnic University of Valencia in 2004, and his PhD of agricultural engineering from the same university in 2007. He actually did also, during his PhD, short internship at the University of California in Riverside. And for his PhD, he worked on the biological control of coccids in citrus. And he joined IVIA after his PhD, where he's working on the development of integrated pest management programs in citrus. And recently, he worked on classical biological control of Triosa erythrae, that is a vector of Hyundongbing using Tamarixia dri. Also, he's working a lot on food supplies for natural enemies in the form of sugars and honeydew. And he's going to tell us more about that work and how they can be contaminated by insecticides and how that can affect biological control. So, Alejandro, the floor is yours.
Thank you very much for the presentation, Cesar, and also thank you very much for inviting me to be here today. It's really a pleasure to be here. And let me start with the presentation. And please tell me if everything works well and if you can hear me perfectly. So now I have started to share the screen. Could you please confirm that you can see it?
Yes. Looks great.
Perfect. So now I'm going to use the laser. Okay, so everything is working. So as Cesar explained, I work at IVIA. IVIA is the research institute that works in the Valencian region to improve the agriculture. And for those of you that don't know Valencia, Valencia is a region in the eastern of Spain, south of Europe. And there we have Mediterranean climate. And the main crop that we have is citrus. In case you don't know, Spain is actually the main exporter of citrus for fresh consumption. So as you can imagine, citrus is a very important crop for us. And of course, I developed most of my career in this crop. So many of the examples that I'm going to explain today are based on citrus, but I think can be extrapolated to other crops. So some years ago, Professor Garcia Maria and myself we decided to rank the main citrus pests that we had at that moment in citrus. Just to let you know, citrus is an evergreen crop. That means the trees have always leaves, and of course they have a great diversity of arthropods. And many of these arthropods, unfortunately, are pests. But they are controlled by biological control agents. As you can see, for example, in this list, many of these pests are in green color, meaning that farmers do not spray to control this pest. And this is because they are controlled by the natural enemies that we have either introduced from other countries or they have naturally adapted to the pest and control them. And most of these biological controls need to feed on carbohydrate sources to meet their energy requirements. So for example, here you can see a syrphids, or below, you can see a parasitoid. As adults, they will need to feed either in nectar, honey, glutation, or honeydew to obtain carbohydrate sources. Otherwise, they will die. For example, you can see in this graph from George Hempel already in 1997, where he showed up that Aphytis melinus, that is this parasitoid of the picture, die just in one day if it doesn't feed on sugars. But if it has access to sugars, the females can live more than 40 days. So you can imagine the importance of sugar sources, of carbohydrate sources for parasitoids or hoverflies. If they do not have access to these food sources, they will die in one day and they will not control our pest. So the presence of carbohydrates in agriculture is very important for our biological control agents. And generally, nectar has been considered the best carbohydrate sources for biological control agents. However, coming back to the, oops, sorry, it doesn't work now. Now it's working, sorry. So thinking on citrus, for example, nectar is very scarce. So for example, citrus trees flower only during a couple of weeks in April. So for example, now, if we go to the field, we will see many flowers in the trees. But after that, the trees are totally without flowers. So the rest of the year, there is not nectar in the trees. Moreover, citrus trees do not have extra floral nectar and do not produce glutation that also can contain sugars. So what happens with the parasitoids if they cannot feed on nectar? This is not something that is happening only in citrus. Here you can see a field of cereals or a field of soybeans. And again, you can see there is no nectar here for parasitoids, for predators, or for bees. But if we take a look close to the plants, what we see is that most of these plants may have aphids, may have soft scales, mealybugs that excrete honeydew. So we don't see flowers, but we can see hemipterans that are excreting honeydew. And this is also what is happening in citrus. If we go into the trees, what we may see is that whiteflies, aphids are excreting honeydew. For example, coming back to citrus, you can see that in spring, after the flowering period, we have many aphid species that excrete honeydew. During the summer period, we have soft scales, mealybugs, and at fall, we may also have some soft scales and whiteflies that excrete honeydew. So honeydew is a carbohydrate source that is abundant throughout the year, not only in citrus, but also in many other crops. I'm sure if you check in blueberries or in soybean, for example, totally different crops, the same will happen because most of them contain some aphids. But do really insects feed on honeydew? Well, this is something that is well known in the case of ants. Most of you, I guess, if you have plants in your gardens or in the house, you may see ants feeding on the honeydew excreted by mealybugs, by aphids. You can see here a couple of pictures in citrus where ants are looking for honeydew. And other insects that feed on honeydew, and it's well known, are the bees. And it's well known because some honey comes from honeydew. So it was well known for honeybees, but also more recently it has been demonstrated that solitary bees also feed on honeydew. But what about natural enemies? What about parasitoids and predators? Well, during the last year, both in my lab and in other labs, we have demonstrated that many different parasitoid species and many different predators, like for example, chrysopids or syrphids, or also more recently coccinellids, and many species of parasitoids are feeding on honeydew. How we have demonstrated that? Well, that's a little bit more complicated than in the case of ants, for example, or of bees that are big insects that you can observe easily in the field feeding on honeydew. So let me explain the case of aphids to explain how we understand that parasitoids are feeding on honeydew. This parasitoid, Aphitis melinus, is a parasitoid of armored scales that you can see in the picture. You can see here the armored scales and the parasitoid Aphitis searching for host to parasitize. Well, this biological control, as I explained before, will live only one day if it doesn't feed on a sugar source, but if it feeds on a sugar source, it can live for more than 40 days. However, his host, the armored scale, do not produce honeydew. So it means it has to search for honeydew or nectar, and also it has to search for host. So does this parasitoid really feed on honeydew in the field? Is it able to feed on honeydew? How we could check that? So let me explain that. What we did in this study is that we went to the field and we measured the number of flowers and the number of honeydew producers in a citrus orchard. As you can see, flowers were totally absent in spring, summer, and fall because we started to sample in May. But what we found in the trees is that, as expected, there were aphids, there were mealybugs, and at the end of the year, at fall, there were some whiteflies. But you can see that there were some differences throughout the year. Well, we measured first the carbohydrate sources in the field. They were mostly hemipterans, and then we came back to the lab and we checked the parasitoids. I'm sorry, it's not working well. Here it goes, perfect. So what we did in the lab is that we collected the honeydews that we observed in the field, but we also collected the parasitoids. With an aspirator, we collected some parasitoids, and there we check and measure the sugar content of the honeydew and also of the parasitoids. And we measure the sugar content, but also we also identify and quantify specific sugars of honeydew using the HPLC. I'm sorry for this huge table, but what I'm showing you here are the sugar contents. You can see here the different sugars that contain the honeydew produced by the whiteflies, some mealybugs, aphids, and soft scales. And what I want to show with this big table is that there are two sugars, earlose and melesitose, that are specific sugars of honeydew. So you can see that these two sugars are produced by all these species of hemipterans, but this sugar is not present in nectar. Here you can see that the rest of the sugars are present in nectar, but not these two sugars. So we use earlose and melesitose as specific sugars to determine whether Aphitis is feeding on honeydew. And here is where the results comes. Here you can see the percentage of parasitoids that had fed either in sugar, in honeydew, or they were starving. They were not feeding on carbohydrate sources, and then they were starving. And you can see that in spring and summer, most of the parasitoids had fed on honeydew. So even parasitoids which host, remember that, their host do not produce honeydew, but they are searching for other hemipterans that produce honeydew to feed on honeydew. But what happened in fall? This is quite interesting because at fall, when the number of honeydew producers was much smaller, you can see that many parasitoids were starving. They were not feeding on sugars. And during that season, we also collect parasitoids in the morning, at noon, and in the afternoon. And you can see how the sugar content of the parasitoid was decreasing, meaning that these parasitoids were not finding food during the day, and likely most of them were dying during the night because they were starving. So during this work, we have seen that honeydew is one of the most abundant carbohydrate sources for beneficial insects, not only in citrus, but also in many agroecosystems, and species of parasitoids, predators, and also is well known that pollinators and ants feed on honeydew excreted by hemipterans, and therefore their nutritional states depend on the presence of honeydew. So keeping on mind the importance of honeydew, we ask whether systemic insecticides may contaminate honeydew. It is very well known, and during the last 20 years, there has been a lot of research that has demonstrated that systemic insecticides that are widely used to control pests move through the plant, arrive to nectar and pollen, and their natural enemies and other beneficial insects that feed on nectar and pollen feed on this contaminated nectar and pollen and either die or have sublethal effects. So we were wondering whether this may happen also with honeydew. So to answer this question, we came back to our main system, to citrus. And there what we did is we use citrus as a plant. As a honeydew producer, we use this mealybug that is called Planococcus titri. This is a citrus pest that actually is not causing too many damages, but it's common in most of the citrus orchards everywhere in the world. And as beneficial insects, we use Anagyrus vladimiri, actually it has changed the name, sorry, and the hourfly Aspheriophora repelli, that is this hourfly that you can see here. Hourflies, for the most of you that are working on biological control, I'm sure you are familiarized with them, but apart of being biological control agents that feed on aphids when they are larvae, they are also pollinators. So many adults are commonly either used or artificially they can pollinate some plants. So by using this hourfly, we are using both a predator but also a pollinator. And as insecticides in this system, we use two neonicotinoids, thiamectosam that moves mostly through phloem and imidacloprid that moves mostly through the xylem of the plant. And this is more or less what we did. We infest the citrus trees with Planococcus titri, the first instar. It developed for a couple of weeks. Then we treated the trees with thiamectosam, imidacloprid, or water. And 3 and 5 days later, we collected the honeydew and checked the presence of neonicotinoids there. So using the liquid chromatography mass spectrometry, we could detect that more than 60% of the trees that we had treated with the neonicotinoids contain honeydew that was contaminated with neonicotinoids, either with imidacloprid that moves mostly through xylem and also thiamectosam that moves mostly through the phloem. So our hypothesis was correct, and we could confirm that neonicotinoids can contaminate honeydew excreted by the hemipterans. But the mere presence of insecticides in the honeydew doesn't mean that it's affecting the natural enemies. So to check that, we offer this honeydew, this contaminated honeydew, to the hourfly and to the parasitoids during 72 hours. And after this time, we measure the mortality of these beneficial insects. Here you can see the mortality of the hourfly 72 hours later, and you can see that it was lower than 10%. However, when the same hourfly fed on honeydew from trees treated with imidacloprid or with thiamectosam, the mortality increased a lot. And in the case of thiamectosam, all the insects had died after this time, demonstrating that honeydew was not only contaminated by the neonicotinoids, but it was also causing the mortality of the beneficial insects that can feed on honey. Similar results were obtained with the parasitoid, but in this case, the mortality was lower and mostly due to the treatment with thiamectosan. So we have demonstrated with this study that neonicotinoids can contaminate honeydew as they do with nectar. This manuscript was published 3 years ago in the journal PNAS, and also the journal Science highlighted these results, and therefore the study had a lot of repercussion in the media, as you can see. And of course, when you have such repercussion and many people know about your study, then some critics arrive. And in our case, we had two main critics. The first one was that we were actually not giving the choice to the parasitoids and the hoverfly to feed in contaminated or not contaminated honeydew. And obviously, beneficial insects in the field may avoid feeding on contaminated honeydew, right? Why do they have to feed on contaminated honeydew if some of the honeydew is not contaminated? So that was correct. This was a good critic. And the second critic was that neonicotinoids are usually mostly as acid treatments, and it is unexpected that these low concentrations of neonicotinoids in seed treatments can reach honeydew and affect the beneficial insects. So those were the critics, and during the last years we have tried to answer these questions. So, do beneficial insects avoid feeding on contaminated honeydew? To answer this comment or this critic, we use the same system. In this case, we use the citrus, the mealybug, Planococcus citri, and as beneficial insect, we use Anagyrus pseudococci, actually it's Anagyrus vladimiri, and we use thiamectosan in this case. And we carry out choice test where the parasitoid could choose between contaminated honeydew and uncontaminated honeydew. We observed the parasitoids during 30 minutes, and we recorded the first selection. And here I'm going to present the choice test. And what you can see here is that unfed parasitoids that had not fed previously on honeydew, choose, contrary to expected, honeydew that was excreted by mealybugs feeding on trees treated with thiamectosan. So actually, what is happening is that is contrary to the previous hypothesis. The parasitoid is not only not avoiding to feed on contaminated honeydew, but it is choosing to feed on this contaminated honeydew. In the case of parasitoids that had fed on honeydew excreted by mealybugs feeded on untreated trees, means parasitoids that previous to the test were feeding on honeydew that was not contaminated. In that case, there was not a clear choice, as you can see here. And if the parasitoids had fed previously on honeydew excreted by mealybugs feeding on thiamectosan, then they choose again the contaminated honeydew. So you can see that parasitoids do not avoid feeding on contaminated honeydew, and even worse, they try or they tend, I would say, to feed on contaminated honeydew. So contrary to the expectations, parasitoids cannot avoid to feed on contaminated honeydew. So our question here was, I guess, the same question that all of you are doing. Why do parasitoids should prefer to feed on contaminated honeydew if this is going to cause their death? So in this case, we don't have a clear answer yet, but what we have is an hypothesis that we are testing right now. We think that neonicotinoids can also affect the bacterial composition. It is well known in other systems that neonicotinoids change the composition of bacteria, and it is well known that bacterial composition affects nectar consumption and also the fitness of not only pollinators, but also biological control agents. So this is happening in nectar. So we guess it may also happen in honeydew, that neonicotinoids may affect the bacterial composition of honeydew and the volatile profile of honeydew, and this may attract the parasitoids to the contaminated honeydew. Well, the preliminary results are showing us that actually this is somehow happening. So what I'm showing in this graph is the bacterial abundance in honeydew from control trees and honeydew from trees treated with thiamectosam with blue color. And you can see that many genus of bacteria are different in honeydew from control and thiamectosam. In fact, we have observed that 16 different bacteria are more abundant in honeydew excreted by mealybugs fed on untreated trees than on trees treated with thiamectosam. And the contrary also happens. As you can see here, some of these bacteria are much more abundant in honeydew from trees treated with thiamectosam. So clearly, thiamectosam or the neonicotinoids are not only affecting the insects, but also the bacteria, and this may change the volatiles. And this is what we are studying right now. Hopefully, in the coming months, we will be able to answer whether these bacteria are also changing the volatiles, and it explains the change of the parasitoid to feed on contaminated honeydew. So that was the answer to the first critic. Let's go for the second one. If neonicotinoids can contaminate honeydew when they are used in the seed treatments. And for that, we move to the University of Minnesota. We went to George Hempel lab, and there we changed totally the system. As you can imagine, Minnesota has nothing to see with our Mediterranean climate. And there we work with the soybean aphid in obviously the soybean plants. And as insects, as a beneficial insects, in this case, we use the predatory mites Aphidoletes aphidimitha, and as parasitoid, we use Aphelinus certus. And as insecticide, we use the insecticide thiamectosam, but in this case, we also use a very common formula for seed treatments. We use seeds coated with thiamectosam, but also with fungicides. So again, we did something similar to what we did in citrus, but in this case, we infested the soybean plants with more or less 15 aphids per plant. And a few days later, we caged the plants. And after that, between 30 and 43 days later, after showing the plants, we collected the honeydew excreted by the aphids. And in this case, we use an ELISA to determine the concentration of neonicotinoids in the honeydew, coming from seeds uncoated, control seeds, seeds coated with thiamectosam and fungicides, and seeds coated with thiamectosam. And as you can see, the concentration of neonicotinoids was 10 times or 20 times higher in honeydew that were coming from seeds coated with thiamectosam in both cases than in uncoated seeds. So between 30 and 43 days after showing the soybean seeds coated with thiamectosam, soybean aphids survived to the treatment, but they excreted honeydew contaminated with neonicotinoids. So again, the neonicotinoids are arriving to the honeydew. But was this concentration enough to affect the natural enemies? So for that, we check the survival of Aphidoletes aphidimitha, as you can see in this graph. And as you can see, the survival of the predatory mites was lower when this predator was feeding on honeydew from the seeds coated with thiamectosam. In both cases, and we didn't find differences between the seeds coated with thiamectosam and the seeds coated with thiamectosam plus the fungicides. So the contaminated honeydew from the coated seeds reduced the survival of the predatory meats, and it was independent of the presence of fungicide. And something very similar occurred with Aphelinus certus, as you can see in the graph. It reduced again the survival of the parasite. So we could answer more or less the two main critiques that we received after our publication. Parasitoids do not avoid feeding on contaminated honeydew, and in fact, at least in the case of Anachyrus, they prefer contaminated honeydew. And at least in the case of the soybean aphid, it can excrete contaminated honeydew that also reduce the survival of some beneficial insects. So during these 3 years after our manuscript, several other manuscripts has been published that have demonstrated that honeydew is contaminated with insecticides. In fact, it has been demonstrated for 5 different systemic insecticides, not only neonicotinoids, but also 3 other different systemic insecticides for 4 different midterm species and with 3 different plant species. And recently, we have published in Biological Reviews a manuscript where you can find some of this information, and we also explain the importance of these findings. And we think that honeydew contamination should be considered in future environmental risk assessment of systemic insecticides in the same way that nowadays is being done with nectar. And with this, I finish, but I want to finish also with an acknowledge. And in the first case, I want to acknowledge Miguel Calvo, because most of the research was carried out during his thesis that he presented last October in the University of Wageningen. And I also want to acknowledge very especially to my colleague, Joel González Cabrera, who has worked very, very hard to try to identify the neonicotinoid or to find the neonicotinoids in the honeydew. And also my collaborators, Dr. Marcel Dick and George Heipel and their labs. Thank you very much. And all my colleagues at IVIA that have worked very hard to carry out this project. Thank you very much. Now, if you have questions, I'm here.
Thank you. All right, so it's difficult to do here on the, but we'll do our best to clap. That was awesome. And as usual, raise your hand if you have a question, or just speak up and ask questions. I see some stuff coming out in the chat, but.
So I have a question if no one is asking now. So great talk, I enjoyed it a lot. One question that I have is because you see differences between the parasitoid and the hoverfly in terms of survival when you feed them with the honeydew with the insecticide, but the parasitoid didn't seem to be affected that much. The survival decreased, but didn't seem to be as dramatic as the hoverfly. And my question is, could you carry experiments to check the fitness of those that survive, of those parasitoids that survive? Was the fitness decrease or increase after they were exposed to those insecticides?
Good question, Alvaro. This is something that we have seen in both systems, in the citrus system and also in the soybean system. And it's quite interesting that in both cases, the Hymenopteran parasitoids were usually surviving more than the Dipteran. Because if you have realized, the other 2 beneficial insects that we used were Dipteran. In one case, it was a hoverfly, and in the other case was a predatory meat, and they were both more sensitive to insecticide. And this make think a lot about the systems that we are using to test the toxicity of insecticides, because we have always thought that the parasitoids are very sensitive to insecticides, but actually what we are seeing is that hoverfly, especially hoverfly, are much more sensitive to insecticides. So I think we should start using a lot syrphids instead of parasitoids to check the sensitivity or the toxicity of insecticides. And also I wanted to mention that I didn't explain all the results here, but we also compare different parasitoids in the case of the soybean aphid, and there were differences even between parasitoid species. So there is always some differences between species, and this is telling us is that for this and for many other studies of toxicity, is very important to study each case or case by case.
I have a question about that difference, and that's really interesting. So if you have an insect that is feeding on the plant is going to have these secondary compounds that is trying to use to minimize. So there's toxic substances in the phloem, irrespective of the insecticide, right? So a plant. I have one question, which is: are there less secondary compounds in the honeydew than directly in the phloem? So how much is there in, pardon me if I missed that right at the beginning, how much nicer is a honeydew compared to the actual phloem?
So this is a very good question. I had a couple of slides before, this morning that I removed because I thought that my talk could be too long. And actually, when we talk about honeydew, something that we need to keep in mind is that there is a huge variability in the quality of honeydew for beneficial insects. So let me explain. Some honeydew, for example, some aphids excrete honeydew that by itself, even without insecticides, are toxic for natural enemies. Okay, so it may happen. Why? Because these aphids are feeding on plants that have some secondary metabolites that are excreted in honeydew and then become toxic for the natural enemies. So this is happening. It may happen. It has been demonstrated that some honeydew increase the longevity of a parasitoid, for example, 1 day, 2 days, but never 40 days like honey. But that was the general idea some years ago, that all the honeydew was toxic because it has no sense that an hemipteran feeds their natural enemies. But what we have seen during the last years is that there is a lot of variability. And for example, in the case of citrus, most of the honeydews are of high quality for parasitoids, especially for parasitoids that are the system that we are working. So yes, some honeydew by itself, it may contain secondary metabolites coming from the plants, but many honeydew has a lot of quality as a nutritional food sources for beneficial insects. Did I answer, Dina, to your question?
Yeah, you did. Although now I have a question that is sort of which you're going to get, the follow-up question on are parasitoids avoiding contaminated honeydew, which is, you haven't given it enough time. If you were to compare I don't know how long, I work with mosquitoes and things like that. So I'm not familiar with how long have people been using systemic insecticides, but if you were to compare a system where this has been used for a long time, as opposed to a recent study or recent system, would there be the evolution of avoidance?
Good question, very good question. I never thought about this evolutionary question. Neonicotinoid has been used for more than 20 years. I don't know if that gives time to the evolution of this avoidance. I'm not an evolutionary biologist.
I mean, it really has to do with Alvaro's question about how deadly is it? Because if it's really deadly, there's strong selection and things can happen. We're talking about insects, right? If it's a so-so selection, then it may take longer. So So I am an evolutionary biologist, so.
So in the case of the, again, I didn't show all the slides. And in the case of the avoidance, what we saw is that, again, there are differences between species. And in this assay, where we check whether the parasitoid can avoid contaminated honeydew or not, we also check that with the syrphid. The syrphid has not co-evolved with the hemipteran that we were using. Okay, so we use the hemipteran that excrete honeydew, the parasitoid that has co-evolved with the hemipteran, and a syrphid that has not evolved at all. The syrphid didn't mind if it was contaminated or it was not contaminated. 50%, 50%, 50%. In the three cases that we studied, doesn't matter the previous experience, it was 50%. In the case of the parasitoid that has co-evolved and knows very well the volatiles of the honeydew of his host, it was a clear difference. It was a clear difference. So this is telling us that a parasitoid that has co-evolved with the hemipteran is affected more by the insecticide. Whether it should avoid, obviously, if it causes a lot of mortality, yes. In our case, we were what we observe is about 50% of mortality. I don't know if that's too much or is too low to avoid the evolution of a trait that makes avoid the contaminated honey.
I'm gonna have, Jacqueline has her hand up, so I don't know if you can see the hands. I usually, if you can call them. I saw Jacqueline and then I saw Kasha also with a hand, but Jacqueline was first.
Okay. Good afternoon. My question was about the honeydew and if it's similar to actually the honey that bees produce and if they have the same amount of bacteria as present. I don't know if you know it.
It's known that honey can contain neonicotinoids, but I don't know if honey from honeydew can contain neonicotinoids. I guess it may, but it's only my hypothesis. It's not something that I'm sure about.
And then I had one more question. I know you said that all the trees weren't treated, but was there a prior treatment to your experiment on that orchard in particular? Since other people do studies.
Sorry, Jacqueline, can you repeat the question? I couldn't hear it clearly.
I know you had treated the trees for your experiment with the insecticides, but was there a previous management done on those orange citrus trees prior?
Okay, so in the case of the citrus trees, they were 1 year old trees, okay, and during it was 1.5 years or between 1.5 and 2 years, and during the last year they were not treated with insecticide except mineral oils that are not systemic insecticides. So they were not treated with neonicotinoids per sure during the last year. And when we bought the trees, we asked if they were not treated. Something that we need to trust, but something that we cannot be totally sure, right? But let me also explain about the soybean aphid, because it was more interesting. In the experiment with the soybean aphid, we used blocks. It was a field from the University of Minnesota, and we used blocks where we had the seeds coated with the neonicotinoids, blocks with the seeds coated with the neonicotinoids and with the fungicides, and blocks without with seeds not coated with neonicotinoids. And I don't know if you realize that in the control we also had neonicotinoids. So Probably, and this is to highlight the importance of the presence of neonicotinoids in the field. It was either coming from the blocks that were treated with the seeds or from previous years, because it is well known that neonicotinoids can remain in the water and in the soil for many, many years.
Thank you.
And Kasia, go ahead.
Hi, I'm Kasha. Thanks so much for your talk. That was really cool. I actually work with spotted lanternfly, and they produce a ton of honeydew because they're really large insects. And we've seen a lot of feeding with like bees and wasps and basically anything else. So it's really cool to see that. So my question kind of piggybacks off of Dr. Fonseca's question to do with the evolutionary aspect of it. So just in general, if the systemic insecticide is meant to kill the aphids, they themselves would potentially be releasing different volatiles to the parasitoids. So could you assume that potentially the amount of parasitoids within that area where those systemics were put on those plants would actually be lower, so they would come in contact with the infected honeydew less often, just because they wouldn't be attracted to the aphid host themselves?
Sorry, Kasia, I didn't get all the things. I heard you very well, but I didn't get all the things.
Sorry, I always talk in circles and I talk really fast. Okay, so the idea...
Let me take notes. I will try to answer.
Okay, I'll try to reword it. So if you have a block that has a systemic treatment on it for the trees, the aphids are going to feed on it. So
correct.
Could they?
And they survive. Some of them will not survive. Some of them will survive, especially some days after the treatment.
Yes. So would they potentially release a different volatile to the parasitoids, basically telling them that they are not a suitable host because they have a weakened system because of the insecticide? So then the parasitoid could then...
Sorry, you are assuming... Sorry, Kasia. So you are assuming that the aphids have volatiles?
Maybe. Yeah,
different?
I'm asking. I don't know. Maybe I'm asking.
I don't really know. It may happen. It may happen that they release different volatiles. Probably the insecticide treatment affect the volatile profile of the aphids. But okay, let's assume that the aphids release different volatiles. Okay.
Yes. So then, would those parasitoids then technically move on to another block that did not have a systemic because they didn't have good hosts in their systemic block because the aphids are sick?
It will depend on the volatiles of the aphid. So
if they have them.
I cannot answer, Kasia. I cannot answer, but if for sure, if it affects the volatiles that the aphids in this case are releasing, it will affect the parasites for sure.
That could be a cool next step as well. You could do like a choice test between, infected aphids and non-infected aphids and see which one's the parasitoids preferred.
Yeah, but that's a very nice one.
Thanks so much again for your talk.
Thanks to you, Kasia.
Grayson.
Oh, hi, hello, let me lower my hand. Thank you for giving talks, very interesting. I was surprised that there was such an impact on the bacterial community in the honeydew. And I'm sorry if I missed this, but did you compare the nutrient composition of the treated and untreated honeydew? Because I think in the neonicotinoid honeydew, they had a lower diversity. I don't know if that means it would be a lower total amount of bacteria. So I wonder if the carbohydrates are the same. And then also if you had thought to look at, I'm assuming these scales have a symbiont as well, which might be affected and might also change the nutritional composition of the honeydew.
Very good. There are actually two questions, so very good two questions. In the case of the symbionts, yes, we check both the bacterial composition in the hemipteran. In this case, it was a mealybug, and also in the honeydew. The bacterial composition within the mealybug, within the hemipteran, didn't change a lot. It was dominated by by a symbiont, and this symbiont was the most abundant one with a lot of difference. 90% of the bacteria identified were this symbiont, and we couldn't find any difference. In the case, so this answers your second question, and the first question is what we are trying to do now, is whether it changed the composition of carbohydrate, and not only carbohydrate, but also proteins. But because usually when we think about honeydew, we think as a food source that is based, or it has mostly sugars, and this is correct, but it also contains a few proteins, and these proteins are very important for the natural endings. So this is something that we are checking right now, if this change in bacterial composition change also the carbohydrate content of the honeydew, and as I said, the volatiles.
Okay, great. Thank you very much.
Nothing that we can answer yet.
Yes.
And Julia, introduce yourselves. I mean, do you have a little background, guys?
Yeah, that would be
Hi,
great if you can introduce a little bit and what you are working on.
I'm Julia. I am a senior undergraduate, and I'm working under Dr. Hamilton and Dr. Hawkins, and I'm doing like a choice study investigating like host preferences in the brown marmorated stink bug. So my question, actually, Dr. Fonseca provided us with some of your papers, like to read up before we had the seminar. And I noticed that the one paper from 2016 mentioned that the presence of ants feeding on honeydew could be an indicator that like this honeydew is most suitable for or most nutritious for the parasitoids. So I have two questions. First, I was wondering if there's any evidence that you found that the ants are being equally impacted, and secondly is and how that influences like the competition for this like ideal sort of honeydew, and also is there any evidence that the parasitoids use like cues from the ants themselves or the presence of the ants in selecting the like most nutritious honeydew?
Wow, Julia, very nice questions, and I'm very glad that you read the papers. I hope you like it. And I think you mentioned three questions. I'm going to try to answer to the first one. Yes, we found this correlation that shows that usually when the parasitoids feed on one honeydew, and this honeydew is better for the parasitoid in the field, we also observe that ants try to attend a lot to this honeydew producer. Okay, there is this correlation. Whether it depends really on the quality of the honeydew, or in other words, if the quality of the honeydew is really doing this, we don't know yet. And whether the parasitoids use the ant trails to find the hemipterans, actually what is happening, and this was published just this year in Proceedings B, so I can share also this publication. What is happening is that this parasitoid, Anagyrus, that is the system that we are using a lot, and the mealybug, this parasitoid, if it realizes that ants are around, because it can see the trail of the ants, they will not avoid the colony of these mealybugs, but they will change their behavior. They will go up to the colony and down, up and down, very fast, trying to avoid ants. So what we see is that parasitoids know that ants are around, they use the ant trails and avoid to be in ant trails long times. So they go and up and down, up and down. This is what I can answer to your question. So it's a partial answer, I would say. Whether the parasitoid use the trail to find the mealybug or the honeydew, we don't know, but we know that the parasitoid can discriminate with or can detect the ant trail. Did you have a third question?
No, that was it. That was great. Thank you so much.
Okay. Thanks to you, Julia.
I have a question that sort of follows up on Grayson's, and sorry, Chelsea, give me 1 second here. I got in here first, which is so these aphids that are feeding on these treated plants and are excreting this contaminated honeydew are obviously resistant to the insecticide that they are being that is being treated. So I'm not 100% of the what's the mode of action of imidacloprid or the thiamethoxam, but well, actually I have two parts to this. One, would you like to collaborate on a project, because mosquitoes also feed on honeydew? And so, so things like a parasitoid, you don't want to kill those, but you certainly might want to kill excessive numbers of mosquitoes. Are you doing that already? And there's actually two levels to this. One, will they die? And two, will, by bee feeding on insecticide-treated honeydew, will they actually upregulate their detoxifying enzymes, making it actually harder to kill by traditional public health insecticides? So it's sort of a two-level question. Are you examining that already? Would you like to examine that? And that's it. So cool talk.
So the first question, I would love to collaborate. I'm not working on mosquitoes. As I said, I'm working in a research institute based on agriculture. So, right. But as I said, I would love to collaborate, and I know that mosquitoes feed a lot on honeydew. Mm-hmm. And regarding the second question, well, you had three questions actually about the tolerance and resistance, because you mentioned resistant, that these aphids are resistant. Actually, there are three things. One, they may be resistant. Okay. Two, they may be tolerant. Some insects are tolerant to some active ingredients, right? So, for example, we have another study where we didn't use neonicotinoids, but we used total different active ingredients, and there the mealybugs are tolerant, so they don't die. Not even one die, not even one. So they are tolerant. Okay. And a third case that it may happen is that, for example, what happened with the soybean is that the concentration decreases. So 30 days later, the aphids arrive. The concentration in the plant is not enough to kill the aphids. So the aphids feed on the on the phloem. The phloem contains a little bit of neonicotinoid that doesn't affect the aphid, or does not kill the aphid, I could say. I'm sure it affects somehow, and excrete the honeydew with the insecticide. So three things: low concentration, tolerance, and resistance. These three different things may occur when they excrete.
But I mean, but it can't be resistance if the chemical makes it all the way to the honeydew. So they have to be tolerant, meaning that they can get the insecticide. Yeah, definition.
Why not resistant?
Well, because so I work with parasites, and so usually resistant means that they don't even go through they don't actually technically get into the insect. So you're resistant, meaning you have a gut barrier. That means that, and again, I'm talking about infectious agents. They don't even get into the insect. Tolerant would mean that you become infected and you don't get sick, right? You don't become sick. And therefore you're like a tolerant person with human malaria would just be transmitting the malaria, will be tolerant to the malaria. So in your definition, what's the difference between tolerant and resistant? We should start there.
Resistant, you have developed it. Tolerant, you have it. Actually, it's an evolutionary question, right? So you are tolerant. So we develop, as humans, we develop an insecticide. You test this insecticide with different insects, and it kills one aphid, but it doesn't kill one mealybug, for example. So the mealybug is tolerant. Okay. So now imagine we develop an insecticide as a humans. The insecticides kill an aphids, but some of these aphids survive and develop resistance. because they have
Okay,
better resistance.
okay. So again,
So that's
it's, you
different
know,
between
guys, it's
tolerance and
always,
resistance.
the definitions are these beautiful things. Okay, so in one case you have, it's a secondary development, and in the first case, you simply don't have the receptor. That's how you're
Correct.
defining tolerance versus resistance. Got it. In both cases, in your definition, so the insecticide will still make it through the system, not kill the aphid or not kill the insect and then go into the honeydew. Okay, okay. Yeah, sorry, sorry about it. And sorry, Chelsea, for waiting. And I see Zoe, so I'll come back to you by email probably. I've really enjoyed this conversation. So Chelsea, go ahead.
Yeah, sure.
Yes, I will spare everyone else my introduction because I had the pleasure of meeting Alejandro during my lab meeting prior to seminar this morning. But I have derived a new question for you. And this is in regards to your study where you were comparing the mortality rates from honeydew that was produced by a plant versus a tree. And I'm sorry, the plant and tree species escapes me. But why do you think the mortality rate was so much higher in the tree honeydew? Do you think it was the insecticide concentration, the amount, the quantity of honeydew, or the nutrition, or like a combination of the three?
Now, in this case was because in the case of the trees, of the citrus trees, we collected the honeydew 3 and 5 days after the treatment. Okay, so that means the concentration was super high. In the case of the aphid, as I was explaining before, it was 30 days later. So the concentration was much slower. That makes the difference between the two studies.
So those weren't the seed treated. That wasn't a seed treated study.
Correct.
That was a spray application.
In the other one, in the first study with the citrus trees, we had actually, I didn't show all the results that we have, but we tested 2 different treatments. We use a treatment that was spray, and if you spray, the insecticide can arrive in 2 different ways, directly to the honeydew or through the plant, if it's systemic. So we did spray, but we also did it by irrigation. So when it's by irrigation, the only way that it can arrive to honeydew is through the plant.
Wonderful. Thank you for answering my questions, and fantastic talk.
Zoe.
Thanks to you, Chelsea.
Hi, my name is Zoe. I am a graduate student in the Center for Vector Biology. I study ticks, a couple different species with a special focus on the population structure of Anaplasma pegastophyllum. And I wanted to thank you so much for your talk. It was, this is kind of obvious, but like the way you presented it and the layout was like very engaging. So that was good for me to see. I had a question kind of going off what you and Dina were discussing with the levels of resistance versus tolerance. With neonicotinoids and aphids in particular, is there any risk at low concentrations of like stimulatory effects on their populations and reproduction?
Whether the low concentration of neonicotinoids affect some of the traits of the parasite of the aphids? Are you asking?
Of the aphids, like I think it's I had to look this up because I forgot the word, but ormoligosis, I think. You know, like where the reproductive rate is amplified due to low concentrations of pesticides.
I don't know. I know it happens with thrips,
yeah,
but I don't know about aphids. So I'm sorry, I should check it. But
oh no, it's okay. I hadn't heard about it with aphids either, but it kind of seems like the type of insect that would be prone to that. So I was just curious if you had noticed anything.
Sorry.
Oh no, it's okay. Thank you. Thanks for the talk.
So I mean, you really sort of, this is really changing everything, like sort of a food web sort of level of interactions. I mean, I just want to sort of understand this. So you for some of these species, you're saying that they don't even have the, I actually would like Zoe, I had to go look up a few things, and nice thing about being on Zoom is that we have our computers right here. So the imidacloprid, I'd forgotten it. It is a nicotinoid, so it affects the nicotinic acetylcholine receptors, which we don't actually use for public health, but there is some conversation about using it for public health. And there is evidence of resistance by simply point mutations in the receptor sequence. But do you see, like, so when you use treated plants, are the populations of the pests lowered?
Yes, in the case of,
Okay,
in the case of the neonicotinoids and the mealybug in citrus, the population decreased a lot.
Okay, so then the secondary effects is that there's still some aphids that are making it, they're producing honeydew, and then you're seeing negative effects on the parasitoids, which ends up creating this sort of bounce where you don't have the, basically you're losing the natural predators, and then you have to completely be dependent on the insecticides. Is that the, okay. But it kind of seems to, depending on the complexity of the system, in some cases, you may still have some parasitoids that seem to be less sensitive. And then you may have some parasitoids that are more sensitive. And so it really is kind of very location specific. I mean, what are you seeing sign of, like, if you measure like really downstream effects in terms of controlling the pest itself, how much does the contaminated honeydew actually affect the overall control of the pest?
That's something, Dina, that's a very good question. And this is something easy to demonstrate under laboratory conditions. And this is why, even though if we go to the field and collect the honeydew, then we have to go back to the lab to check the mortality of the parasite. Why? Because then in the field, what we are observing would be the effect of honeydew, effect of nectar, the effect of contaminated water, the effect of contaminated leaves. So there will be many different effects going on. That's why we need to take out the honeydew of the system, bring it to the lab, and check the mortality. The important thing of our finding, in my opinion, is that so far it was considered that these systemic insecticides were contaminating only nectar, right? And pollen. And nectar and pollen, most of the crops, nectar lasts a couple of weeks, 1 week, 3 weeks. But in the case of honeydew, we have honeydew throughout the year or throughout the season. There are always some aphids, a few of them in some plants. So that's why I think our finding is so important. How to measure the importance of this finding? This is going to be hard. We have several ideas, like using caged plants, but to tell you the truth, we still couldn't do it.
No, it's hard. I mean, it's sort of the epidemiological effect, the downstream effect on it's always the hardest part. But this was really an interesting talk. And any other questions, anyone?
I got one quick one. In one of your slides, when you were talking about the neonics, you said that you used a couple of other systemic insecticides. I'm curious what those were.
One was pimetrocin.
Okay.
The other one was pimetrocin and metrocin. Let me check the other one, so I'm not talking by, and I don't. Another one that has been demonstrated not in our lab but in another lab is spirotetramat. So
okay,
spirotetramat, pimetrocin, and the other one is here. Dun, dun. Pimetrocin. and flonicamide. Sorry. So 3 of them, spirotetramat, flonicamide, and pimetrocin has been already demonstrated that they can contaminate honeydew.
Okay.
Now, I was
Thank
just checking the tiamethoxam. Is it also a neonicotinoid?
Yes, thiamectosam and imidacloprid are 2 neonicotinoids. In the case of thiamectosam, is one of the most toxic neonicotinoids.
Right.
So, for example, thiamectosam and imidacloprid has been banned here in Europe. So they can be used only under greenhouse conditions because of the bees.
And actually, that was sort of like in closing. I mean, we have a lot of graduate students and undergraduate students. They're all very good. Great questions. Thank you. So in your experience, I mean, you basically were put in the spotlight by these experiments. Can you just share the experience and as a scientist, how you survived it?
How do I survive to what? Sorry, I didn't
Well,
get
the
the question.
experience of basically being put, I mean, you were in the spotlight. You had, I'm sure, newspaper, reporters. It's just that part. I mean, we're talking science, it was great discussion, but there's also the social aspect of being a scientist. Would you just share that experience?
Yes, of course, I can share that experience. It's always very nice to see the recognition of first of all, of your colleagues and the society also. But these kind of results are also quite challenging for people like us that are working in agriculture, because we know that we need to use insecticides to control some of the pests. But at the same time, we use this overuse of insecticides that we are totally against this overuse of insecticide. And then we need to understand how this overuse of insecticides are killing our natural enemies and are destroying our biological control. Because, for example, here in citrus, we see that when they used to treat with neonicotinoids in spring, then in summer they had problems with many pests. Why? Now we are discovering why. Because these neonicotinoids remain in trees for many, many months, and many insects, beneficial insects that are feeding either on honeydew or on their own plant, they die. So these kind of results are always a bit complicated to explain to the farmer and say, look, if you spray with this insecticide, you are going to kill your pest, but some of your pests are going to survive and are going to kill your natural enemies. So that's a little bit hard. So on one point, you have the recognition of your colleagues because you have published your data very well, and the media is asking you, so you are in the journal, but at the same time, you have to go to the farmer and say, we have to find another solution, and sometimes you don't have an alternative solution. And that's part is hard.
All right, well, thank you very much. And Cesar, I hope you're still there in Peru. Really, it's great. Thank you for a great talk, great discussion, and thank you. And
Cesar, do you have any closing comments?
yeah,
I'm here. Thank you. Thank you, Alejandro. I appreciate it.
all right.
Thank you very much to all of you.
I'm sorry?
Are we still meeting with the students?
Well, I mean, we basically had a half an hour for conversation,
Oh, okay.
so I think we're good. I mean, unless
We're good.
everybody has to actually and guys, I'm going to be sending an announcement. Check your emails because this was the last seminar of the semester.